Hello there, welcome to our channel. Today we'll be discussing the embryology of the cardiovascular system. However, not to bore you and keep you glued to your screen for longer than it should, we found it best to give you the lectures in sections to help understanding and assimilation. So let's get to it. As the fetus develops, the fission, which is the main mechanism of nutrient and oxygen distribution in the prior weeks, become inadequate to continue this function, making a system which will provide and serve as an effective medium of transportation or distribution necessary. The development of the cardiovascular system begins at about the third week of gestation, coinciding with the development of the uterine-placenta circulation. The system is mesodermal in origin. However, there are special group of cells which are directly involved in the development of the heart, called the progenitor heart cell. The progenitor heart cells lie in the epiblast just adjacent to the superior end of the prometheus tree. Subsequently divided into two groups based on their final destination after migration, we have the primary heart field and the secondary heart field. The primary heart field are composed of cells which migrate through the streak into a lateral plate of the mesoderm, forming a horseshoe-shaped cluster of cells. The migration occurs between day 16 to 20 of gestation, and upon arrival here, patterning of these cells or group of cells as well as laterality of the whole embryo occur simultaneously. The process is mediated following a signaling cascade, with serotonin being the key molecule. The derivative of the primary heart field will include the atrial, or we say muscle, as well as the left ventricle. The secondary heart field are composed of a group of cells which upon migration come to reside in the visceral mesoderm, ventral to the pharynx. They appear much later at day 20 to 21 of gestation, and this group of cells as well undergo patterning and laterality. We can say that they contribute to the rest of the heart, as its derivatives will include the outflow tract, where we have the truncus arteriosus and the cornus cordis, the right ventricle, and even some part of the atria. Normal heart development is mediated by the expression of the NKX2.5 gene, which is regarded as the master gene for normal heart development. This gene is expressed following the combination of the secretion of the BMPs by the anterior endoderm underlying the progenitor cell, plus the inhibition of the WNT expression. Once the primary heart field has been established, the induction of the cardiogenic region is initiated by the underlying endoderm as the primary heart field, is acted upon by the fibroblast growth factor 2 to become hemangioblast and then blood vessel. The other protein involved is the vascular endothelial growth factor, which acts via two different receptors to give us this endothelial 2. The horseshoe-shaped island of the primary heart field gave rise to two ventral longitudinal vessels which are termed the endocardial tubes. In addition, other blood islands appear medially and bilaterally and are as well induced to form a pair of longitudinal vessels that form the dorsal aorta. Initially, the cardiogenic region is anterior to the oropharyngeal membrane and the neural plate. However, following the rapid growth of the brain, consequent to the closure of the neural tube and the formation of the brain vesicle, plus embryonic cephalic folding, the oropharyngeal membrane is pulled forward, while the cardiogenic region consisting of the heart and the pericardial cavity moves inferiorly to the cervical region and then finally into the thoracic region. Simultaneously occurring is the lateral embryonic folding, with the two endocardial tubes merging to form a singular heart tube. Fusion of these tubes begins cranially and then extends inferiorly, however sparing the caudalmost end. The tube, however, remains attached to the pericardial cavity as its dorsal side by a fold of mesodermal tissue called the dorsal mesocardium. The tube continues to elongate with the addition of cells from the secondary heart field at its cranial end. This lengthening is essential for normal development of the right ventricle and the outflow tract. Preliminarily, the fused continuous endocardial tube can be divided into four parts. Cranial to caudal, we have the truncus arteriosus, the bulbus cordis, the ventricle, and the atrium. thus receiving venous drainage via its caudal end and pumps blood through its cranial end into a dorsal aorta. Further development occurs with the addition of the myocardium, which is derived from the surrounding smoothing mesoderm. The myocardium thickens more and begins to secrete an extracellular matrix rich in hyaluronic acid called the cardiac jelly. The cardiac jelly separates it from the endothelium. The epicardium is got from the proepicardial organs, which are a special group of mesothelial cells at the base of the heart, that is, the external surface of the sinus venosus. This group of cells undergoes cell proliferation and spread cranially to cover the entire heart tube. Also, there is the appearance of a transverse pericardial sinus as the midsection of the dorsal mesocardium degenerates, thus connecting both sides of the pericardial cavity. The heart is now suspended in the cavity by blood vessels at the cranial and caudal end. So moving forward, the lengthening of the heart tube is essential for the next step in heart development, called the cardiac looping. Prior to this process, the primordial heart tube extends beyond the primordial pericardial cavity. The process begins at around day 23 of gestation, as the cephalic portion of the heart tube bends ventrally, caudally, and then to the right, as the atrium moves dorsocranial and then to the left. The process is completed around day 28 of gestation. Upon completion, the heart is now fully incorporated in the primordial pericardial cavity, with visible local expansion and narrowing in various portions of the heart, most noticeable at the atrioventricular junction and the bulboventricular junction, which give rise internally to the atrioventricular canal and the primary interventricular foramen, respectively. At about the distal half of the bulbus cordis, which is narrow compared to the proximal half, this part later gave rise to the trabeculated part of the right ventricle. However, the proximal part, which is termed the cornus cordis, gave rise to the outflow tract of both ventricles, while the truncus arteriosus formed the root as well as the proximal part of the aorta. Given the complicated path by which the heart develops, a normal or flawless heart development is not always guaranteed. So let's dive into the section of what could go wrong. The establishment of laterality during gastrulation is integral for normal heart development owing to the cell specificity to how they contribute or patterning of the right and the left side of the heart. This process requires the signaling cascade of which serotonin, as mentioned earlier, is key in initiating this pathway. This pathway ultimately leads to the expression of the PITX2, which is the master gene of left-sidedness. Point to note is that serotonin is known to be concentrated on the left side of the embryo or fetus. This process occurs at about day 16 to 18 of gestation and thus critical for normal heart development, as disruption could lead to dextrocardia or even situs inversus. We could also see atrial and ventricular isomerism, where there are similarities between both atria and ventricles, thus losing the normal left-right differences. Another heart defect is the inversion of the heart chamber, which is characterized by the reversal in the characteristics of the atria and the ventricles. Other heart defects that could manifest with the disruption of this process will include double outlet right ventricle, atrial septal defect, as well as ventricular septal defect. The importance of laterality in normal heart development is further buttressed by the teratogenic effect of the antidepressant of the selective serotonin reuptake inhibitor class that has been linked with increased heart defects, which is used in pregnancy for the fetus. When the lengthening of the heart tube is inhibited, either by a destruction of the secondary heart field or by the inhibition of the signaling pathway, such as that which leads to the expression of the SHH that is involved in stimulating cell proliferation in the secondary heart field, an array of outflow tract anomaly or defect could be seen. Another mechanism by which outflow tract defect or anomaly could manifest is if there is an inhibition in the noise signaling pathway, which is known to act through its ligand JAK1, that is responsible for upregulating fibroblast growth factor in the secondary heart field, that in turn then regulates cell migration and differentiation of the neural crest cells, which is essential for outflow tract division, as well as development of the aorta and patterning of the aortic arcs. With this, we've come to the end of this video session. Hopefully you enjoyed it, and we look forward to catching up with you in the next. Please remember to give this video a like, subscribe to our channel, and click on the notification bell to keep you in the loop for our video updates. Remember to share this video with your friends and colleagues. Till next time, thank you for watching.